to the last two of the lectures, which where I want to discuss with you some of the very modern techniques which in future will be part of the regular cell culture lab. So, if you remember the last class where we talked about integrating microtunnel on top of the microelectrode arrays. So, I did not kind of spend time on how the microelectrode arrays look like. So, it is something like you have a small chip like this. It's kind of, you can say, something of this size, this is probably this is the kind of size. If I put two of my fingers here, like maybe a little bit more, that is it. That is the size of a microelectrode array, and a microelectrode array has these kind of the electrode which are embedded. So, what you saw in the picture in the last class is something, there is a microelectrode array, on top of that those channels were being placed like this. Like this, something like this, okay. Ensuring that, and of course, you have a partition somewhere or other in between these two. So, if you wanted to study the connectivity between place A to place B, so you place the neurons of place A of the brain on this side, A side, which is, whereas you place the neurons from the place B of the brain on this side, and allow the single neuron processes, because the diameter of this tunnel is around 2.5 micron, which essentially could accommodate, if you see a typical brain neuron. So, it is something like so the cell body is around 30 micron, whereas the cross sectional area is approximately 2 to 3 micron. Okay. So, essentially one process can go through, or maybe two, but the thing is that in real life this 2 micron includes the myelinating oligodendrocyte. But when you are doing a chronic culture like this, if you are not doing myelination, then actually in a tunnel like this you could have 3 to 4 or maybe 1 to 2 axon traveling through. So, something like this, okay. And of course, you have the neurons of this part sitting here. So, essentially this microtunnel technology can give you an idea. Say for example, if this is the brain what we are talking about, and so you wanted to study, say for example, a connectivity between this part A and this part B, or link between say C and D, or from A prime here to another tissue or another neuron at say somewhere in the spinal cord SC3. So, you can study all these connectivities by using these kind of say electrical system coupled with a small tunnel technology or microchannel technology, whichever way you feel comfortable to express it, microchannel technology. Along with it, you could have several other microelectromechanical system coupled with it. So, in a way, say for example, I create a situation, say for example, I say that I wanted to study the connectivity of say parasympathetic neuron, okay, parasympathetic neurons with adrenal gland of kidney, okay. So, the on top of the kidney of the adrenal gland. So, there is a connectivity between parasympathetic and the adrenal gland. Now, how I could study it in a real situation in a real animal, that will be a very cumbersome process. It will take forever, and you will not be able to understand all the basic mechanisms. So, how you can do it? So, you take the adrenal tissue, or of course, whether the cortex or the medulla, which part you want to do, because the adrenal, if you look at the adrenal, it has a cortical region, it has a medullary region. Okay, it is the outer periphery and it has an inner core, and based on that they have different functionalities, right? And you have these parasympathetic neurons. So, now what you do, you isolate the parasympathetic neuron and innervate the parasympathetic neuron with the cells of the adrenal in a, say, multi-compartment chambers. So, there are several multi-compartment chambers which are being developed all over the world, and the genesis of all these things starts with the Campanot chamber, which I talked to you in the previous class, which was the very first of the multi-compartment chamber. And now, following up on another aspect of the last class, what we talked about when we talked about the microchannel technology coupled with microelectrode arrays, and today I will request to upload the particular paper which I have discussed in the previous class. You will observe something interesting, that in a 2.5 micron channel there are 4 different population of electrical activity which emerged. It seems the neuron at one point, while getting connectivity with another part, so within hippocampus, say for example, if the connectivities are happening, so between say dentate gyrus to say CA1. So, it has been observed that the CA neuron crosstalk with dentate gyrus not at a uniform spike frequency. Instead, at least it is clear there are at least bare minimum 4 different kind of spike frequencies with which they could talk, that too in an extremely chronic in vitro system. But in an in vivo, probably this number may go up. So, that means different connectivity, say for example, I am trying to establish connectivity between A and B. So, essentially the A population has several subtypes, A1, A2, A3, A4, A5 likewise, and similarly the B population has several subtypes which are B1, B2, B3, B4, B5, and there are connectivities like say for example like this, or say like this, or say like this, and based on these connectivities there are different spike frequencies as well as the speed. So, it means these connectivities are function of different level of resistances which are created, and that could lead to different form of, now if I translate it in terms of a circuit element, then you will realize that how the biological system transfer information at its own whims and fancies. And as a matter of fact, the future of much of this computation will lie in understanding how these things are happening in vitro. Second, on such setup, you can improve the electrical activity. It could be much more closer to the real life situation if some way or other you can incorporate the glial cell into the picture, which will create the insulating cover what these cells are needed in order to maintain the fidelity of the signal. Okay. But these are some of the techniques which will take us a long way. These are not so easy because you are trying to rebuild a system outside. So, this is essentially falls under the purview of in vitro developmental biology or in vitro development, because you are seeing the development outside the system. So, what you are essentially doing is you are ripping the system out. Say for example, this is a actual system which has different components, say A, B, C, D, E, F, G, H, I, J, K. Now, essentially what you are doing, and say for example, there are multiple connectivities between these different things. say for example, it is a complex connectivity which is happening. Now, what you are trying to do is, you wanted to say for example, study the connectivity or the crosstalk between say point B and point J. Now, what you are doing is that you are isolating point B, you are isolating point J, and you are rebuilding the connectivity. Now, if you look at even here in the most simplistic note, so this is B to K, K to D, and probably D to J. So, there are this signal in the real situation, the signals are with say K, D, and then coming to J. But now you are creating a direct connectivity. So, this direct connectivity will create certain kind of resistance or certain kind of spike frequency between B and J. So, now in a real life the data won't support. Real life will say that there is some difference, there is a lacuna or there is a discrepancy in the data. How to justify that? You can only justify that when you now introduce these components. So, you get the, so you have the B now, then you have a connectivity with K, it has a connectivity with D, it has a connectivity with J. But that also does not tell you the whole story, because you have to realize here you have connectivity of J, influence of J from F. You are realizing, and similarly I, which is sitting here, may have an influence on J. We do not know those connectivity, but now looking at a whole brain, probing electrodes at different places in vivo may not give you those nitty-gritty details of resolution what you are looking forward to. The only way you can approach the finer connectivity or how this computer architecture of the brain or other parts of the body works is when you can rebuild the systems in vitro, and that's where is the growth or the beginning of the concept of biohybrid system. And this biohybrid system is essentially you are marrying the electrical and electronic components with microfabrication technology along with cell culture, chemistry, surface chemistry, and a whole plethora of different technologies in one platform. So, this is a very, very emerging area of cell culture, and this is where the future lies. Some of the labs which I will request you people to visit will be, at least in a virtual reality, will be Professor James J. Hickman's lab. They are the pioneer of developing a series of biohybrid systems, and most of the genesis are from Bell Labs. It all started there and kind of influenced people later. Then you have Professor Bruce Willer, Professor Gregory Brewer, and in the same token you can look for the people who have worked in Germany on field effect transistors, IFETs, ionic field effect transistors, okay, and microelectrode arrays. So, these are some of the, especially Professor Bruce Willer, Professor Hickman, and Professor Brewer, these are some of the very pioneering individuals who laid the foundation stone of looking at things from a very, very engineering perspective, rebuilding the systems, rebuilding the system on a small microchip, and then studying biology. And it's not that all the time you needed to answer a question of biology. You may actually dream of using biological component for the future level of computers. Okay. So, using biological tools for developing computers of future, or you can call them biocomputers or biocyber computers, whatsoever you like. So, the point I wanted to make, like these are the aspects which are not dealt, yet these are not textbook. It will take at least another 10-15 years before these become textbook, because probably we needed to write new textbook, because these are the emerging technologies which will be governing the field of cell culture, the field of cell culture technology, the field of drug discovery in the years to come. And not only that area, the ultrafine computing at the level of single axon level, single neuron computation, nerve to muscle computation, and a distant far reaching goal for a distant future. Could we have computers made up of proteins? Could we have computers made up of cells? A far cry, a dream which mankind has been seeing or dreaming for a long time. So I'll close in here with the word of hope that someday, somewhere in a distant future, these technologies will make a difference in our life. Thank you.